Yes, doctor.
Okay, so a very good afternoon. Okay, again, I would like to welcome students from Universitas Asia Yogyakarta who is majoring their MLT in biomedical science, MLTs, and also my own student from University Kuala Lumpur Mestek in Kajang, Malaysia, who is undertaking 4-year biomedical science program. And in their 4 years, they learn various subjects, and one of the subjects they learn is parasite under microbiology. And at the same time, I would like to welcome my master student, my second master student, Nurul Nadia, and she is working on parasite, but what's your research? She's trying to see kalau antigen dari parasite tu boleh treat the colon cancer. So she's working on that side lah, a bit on biotech. So my name is Dr. Nisha. So this is our title today, Molecular Diagnostic Technique for Parasitology. Kalau sepanjang pengajaran saya, if you have any question, you can stop me. I will talk in Dwi Bahasa, which is I think quite okay for this topic. Okay, so sebagai introduction, we know that parasite is an organism that lives in or on an organism yang diambil makanan. So we know that it gets food and so on. There are 3 main classes of parasite. Example, we have protozoa, helminths. Helminth ni cacing, you ada cacing pita, cacing bulat and semua. And one more is ectoparasite. So are you familiar with all this? Like for example, you have blood-related parasite, mainly we learn on malaria. Aside from that, you have ectoparasite, kutu, hama yang hidup di atas badan manusia. And gastrointestinal parasite, parasite yang dijumpa di dalam usus manusia. Yang ni kita boleh pecahkan pada 2 bahagian dah, 2 kategori. One is cacing, another one is protozoa. Protozoa tu macam cyst, giardia, blastocystis and so on. Okay. Next. Okay, type of parasite, as I explained, you have protozoa single cell. Example ialah Entomoeba histolytica. Kedua, ectoparasite. Example, those yang which actually live above our body. And helminths. Okay, helminths example, cacing. So are you all familiar with these, students? Have you all learned before?
Ya, doktor.
Okay. So apakah kepentingan kita belajar cacing atau parasite ni? Actually dia boleh membawa kemudaratan pada our body lah. It can bring, am I still sharing? Wait. Okay. Can bring kemudaratan pada tubuh badan kita. For example, it can cause rashes, weight loss, sakit perut, anemia, sleeping problem, fever, death if in severe condition. Severe, there's a mistake here. So disease bermaksud various disease are caused by parasite. Parasite can be either cacing dia cause ke, malaria ke, ataupun serangga. So how we know seseorang pesakit tu kategori mana yang dia dapat penyakit ni? If jangkitan kuman parasite adalah kurang, macam just ada berfew telur trichuris, then mungkin dia akan dapat just sakit perut, cirit and so on. Tapi if jangkitan mereka is high infection, Kalau kita we refer to World Health Organization punya reference, kita boleh kategorikan infection dia ke tinggi, sederhana atau rendah. Kalau rendah, you makan ubat cacing lah, ubat Xantel macam tu, okay ready. But kalau kita dapat infection yang tinggi, high infection and so on, then you will have more severe clinical symptoms. Clinical symptoms kita agak teruk lah. Macam anemic, sleeping problem, fever. Yang ni kena admit to the hospital or you need to see the clinician for long term of medication. So that's something about that. Okay, let's try this. Then who going to tell me? What is this? Tembak lah. What is this? This one cacing famous. Very easy. Who can tell me? Dia Permata, are you there? Dia Permata Sari? What do you think this worm is?
Is it a tapeworm?
Sorry? Yes, it's a worm. Yes, Umaira, what do you want to tell? Ascaris. Ascaris. Ascaris adult worm and ova. Okay, this is Ascaris. This one pula. Telur ni cantik. You cannot forget the shape.
Trichiura.
Yes, very good. Pandai dah. Okay, Trichuris adult worm and ova. If you see, Trichuris trichuria ni cacing dia nipis compared to this, compared to Ascaris. Ascaris, it can go up to 15 to 20 cm. Pembaris pendek kita ni. Kalau yang ni, cacingnya sangat nipis, tapi dia boleh membawa kes colitis yang teruk. Dia akan membiak cepat sini. So this is Trichuris trichuria. Okay. This look like an alien, very famous to give anemic. Dia cepat hisap darah. Kalau wanita yang mengandung, quite high risk to get abortion, miscarriage. Apa yang ni ada gigi-gigi ni? Cacing ni if you tak pakai slipper, tak pakai kasut, cepat dapat. Anyone wants to guess? Puspita Ayu, any idea from you? Shinta Devi, all new name.
Tanya's species?
No, Tanya. Look at the plate, the planar and plate. From the name is actually here, the Necaratus and Suramada. Tinia is actually you find from animal source. So they are not under here. Tinia dia macam, kalau Tinia you tengok dia tak ada, they don't have a wakul here. Dia padat, cacing dia padat. Tapi kalau ada lubang macam ni, hookworm. Hookworm, larva and ova. Ankylostoma duodenale, Necaratus americanus and so on. Okay, so cacing tambang yang sudah dibuahi. Okay, thank you. Maybe the terms that you use there is different lah. Maybe in bahasa Malaysia or Indonesia, I'm not very familiar, but it's generally we call it hookworm, Trichuris, Ascaris, and so on. Okay, as long we know what is the worm, should be okay. So now, okay. What about this? Now ni pula malaria. Any idea what is this? Kalau tengok RBC ni normal, RBC ni dah bengkak macam besar. This one looks sekata. So, okay. This one we call it Plasmodium ovale. Kalau Plasmodium ovale and also Plasmodium vivax, dia memang kalau RBC yang infected, dia memang besar. Besar teruk-teruk compared to normal RBC. At the same time, bila kita tengok dekat kalau dia nampak RBC dia sama saiz je, most probably is actually falciparum. Kalau falciparum, RBC dia semua saiz sekata, yang normal dan infected. So that is how we identify. Okay, so ini ialah dalam thin blood smear. Okay, have you all learned thin blood smear? Dah belajar ke? Thick dan thin? Thin blood smear, you bubur dekat slide one drop of blood dan you tarik. Yes, okay. Yes, actually you can unmute and mute yourself if you want to tell answer. Okay, good. Okay, now let's go through our main lessons. Okay. So I won't go too deep lah. Itu just like I want to a warm up lecture, a few slides. Okay. So now kita we direct go into the main topic of discussion today. Common technique in parasite diagnostic. Okay. Kalau kita tengok, kalau kita like we are more to laboratory science kan. Even my PhD we more to lab. So we will be more familiar with microscopy technique, molecular assay, serology, culturing method. These are all scientist work. Kita lah ahli-ahli sains. So diagnostic ni ada dua. One, in lab base. Lab base bermaksud MLT akan kerja sini. Biomedical science also okay. Microbiologists can work here and so on. Some yes, biotechnologies. At the same time, ada juga cara lain, which is clinical based technique. Clinical based technique, apa yang paling perbezaan yang ketara ialah clinical based technique ni, mesin-mesin yang ini, the equipment that yang mereka guna directly touching the patients. Endoscopy, colonoscopy, ultrasound, MRI, CT scan and so on. Macam manakah kita buat ni? So for example, yang ni perlukan medical doctors, MD, MBBS, they need to be trained. You need nurse and so on. Okay, kalau scope macam ni, either you put from the anus, from the throat. Like tadi one of your friends say, right, Tanya species. Tinea species can be detected with colonoscopy. Sebab dia memang padat, dia lekat kuat-kuat dekat our usus. So they will go up to a few meter. Colonoscopy can. That is a camera they're putting down. Ultrasound. Ultrasound, I think we are all quite familiar. They use this to see the baby's movement, guna untuk tengok cancer development, that is non-communicable disease. Tapi untuk parasite also, you can use to see whether the parasite infection has give a patient some komplikasi. MRI, yes. CT scan. Yang ni dua, kita boleh guna, especially CT scan. Have you learned about Naegleria? Cacing yang, the parasite yang directly makan otak. Yang dijumpai di air, contaminated water like waterfalls and so on. Ada satu parasite, kita panggil Naegleria fowleri. Parasite ni, dia direct masuk hidung. Hidung. Okay. And directly dia akan pergi ke brain. Dia makan tisu-tisu di otak. So kita akan gunakan CT scan lah to see cyst development in the brain. That is something. Okay, so this is clinical base. So we need medical doctors, nurses to do this. And radiographer, yes, radiographer and so on. Okay, so this is that. Okay, jom kita pergi tengok more detail on laboratory, which is more related to us. Okay, semua dah familiar dengan mikroskopi ke? Are you all good in microscopy?
Ya, dokter.
Okay, tell me something about microscope. Ada berapa kanta ya? Ada berapa objektif mikroskop? Yang paling rendah ialah apa? 4 times?
Yes.
Okay, 4 times. Lepas tu 10, 10, 40.
3 doktor.
Yang
10, 100.
okay, 10, 100. 100 apa kespecialan dia? The specialty of 100. 100, can you just see? I think you all can open your mic and talk. It's easy for me to see when you all type in message. Susah nak tengok.
Jika menggunakan lensa 100 harus memakai minyak imersi.
Yes, you're right. Kita kena gunakan di oil immersion. So that is something about that. So I'm sure you all dah familiar on this. Okay. Molecular base assay. Anyone have done PCR? Ada sesiapa sini buat PCR? Tapi dah belajar lah. So we'll look into this today. Serology. Serology assay. Sekarang masa zaman COVID ni, dia macam dah jadi very familiar lah. Ahli yang bukan dalam field of science pun semua dah buat. Especially your ujian calitan, air liur, nasal swab. Semua ini adalah a part of serology. Example, kita ambil serology tu dalam kit dia sudah ada dia punya antibody. Jadi bila kita ambil calitan air liur ke atau nasal swab, kita put with the buffer, then dia akan detect lah kita positive for COVID or not on. Okay, untuk parasite how we use? This is common untuk blood related parasite. Untuk malaria, for example, we have pan malaria test kit. You take our human blood, okay, kita spin, we take the serum and we put in the kit. Sama saja macam how you do your COVID test kit lah. Kalau dia ada 2 band, okay, patient is positive. Tapi kalau positif, you tanya, okay, species mana ya? If 5X ke, that one quite difficult to say. Yes or no, yes. Kalau dia ada infection ke tak ada, boleh informasi tu boleh didapat dari serology. Tapi, if you want to go specific untuk species mana, that one either molecular, microscopy, atau culturing. Okay, so that is something serology. Culturing pula. Apakah dia culturing? Culturing method is proses untuk membiakkan parasite tu di makmal. Kita ada cara tersendiri. Okay, di makmal saya, we actually have cultured Ascaris lumbricoides. Sampai kita ambil dari tanah dia, culture, guna sikit bahan kimia lah, dan kita dapat tengok until dia molten the 4th stage. So siapa nak guna cara ni? This usually postgraduate student will do this for their research. For them it's more important lah. Maybe they want to take the antigen ke, nak buat PCR ke. So culturing method they will do. So, molecular based assay pula, this is something really related to DNA. As we know, our molecular genetic code in human is mainly DNA based. We have A, T, C, G. So each these 4 codon, and molecular base is very, very accurate. Dia macam sensitif sangat. And cara ni juga digunakan masa pandemik untuk diagnose seseorang itu have COVID positive. Those days, I mean, still we are using, but not as much lah. Now we are just focusing on serology. Tapi kalau you nak terperinci sangat, we use molecular base assay. Sampai sini okay ke? Anyone ada apa-apa soalan? I'll go slow. We have a lengthy 2 hours.
I think not yet.
Yeah, can you speak louder, student?
I think not yet. No.
Oh, nothing. Okay. Okay, so you have nothing, so I have something. Okay, student, kalau I nak tengok cacing Ascaris, cara mana yang paling senang ya? Pada pendapat anda. Cacing Ascaris.
In laboratory based technique or clinical based?
In laboratory. Clinical we don't because it's more to not our field. But I just want to tell you overview. So in laboratory based technique, kalau seseorang itu ada Ascaris, cacing Ascaris, cacing bulat, molecular, cara mana pendapat anda yang paling very easy, rapid?
Microscopy.
Yes, very good. Very smart. Okay, microscopy. Sebab you know already the size is so big. In fact, untuk Ascaris kalau cacing tu you tak payah mikroskop pun. Kalau dah tengok pun dalam bekas kita dah tahu. Tapi kalau nak tengok ova dia, the eggs, trichuris Ascaris, you can just see in the microscope. So microscopy is the best. Okay. Kalau Giardia? Giardia duodenalis. Dah belajar kan Giardia? Giardia ni dua boleh lah. Microscopy atau molecular. Serology also can. Tapi kit dia tak famous sangat. Culturing not lah. Usually kalau Giardia, kalau trophozoite senang tengok di mikroskop. Kalau cyst, kadang-kadang kita confuse dengan lain artifact ke. So you can rajin lah buat molecular assay to confirm. Okay, that is the thing. Kalau hookworm, which one you feel laboratory based technique yang paling cepat? Molecular. Sorry, this one. Microscopy. Untuk hookworm pun is microscopy sebab dia cacing yang panjang. You can actually see. Tapi maybe bahagian mulut tu you tak clear. So if you're not clear, then you are confused. Maybe dia necrotus atau ankylostroma. That one you kena buat PCR. Sebab cacing ni ya. This one. Cacing ni telur dia macam ni. Di mikroskop telur dia I think kena mute participant kena mute I think you all there's background noise. Okay, tak apa. So this is the telur. So telur ni agak mudah lah nak tengok dalam slide. Tapi kalau I tanya, oh good you got hookworm dah, tapi I nak tahu spesies yang mana. Then you have to do a PCR. If from here, kalau cacing, kalau your laboratory skill memang dahsyat lah. If you really can see this, then you can tell either duodenale or americanus. The only thing is cutting plate dia different sikit. That's the difference. Okay, but this one, case dia agak teruk. I read in Indonesia also the rural children memang they have a lot. Tapi kami di Malaysia kita punya problem memang orang asli kita ialah Ascaris dan Trichuris. Ini kurang sikit. Sebab di tanah di pasir di Malaysia, this egg they can't survive. Our soil lah, condition of pasir di sini ya. This one tak survive, tapi ascaris and trichoris memang tinggi. Okay. So let's go. So then we will discuss, continue to discuss about molecular based assay for parasite diagnostic. Okay. Molecular based assay ni memang is very good, memang like rapid, effective, sensitive, specific. Okay. Tapi in bidang parasitology, microscopy examination is still considered the golden standard method for diagnostic of parasite disease. Golden standard meaning this is the main thing. Kalau anyone, if you pergi nanti nak pergi intra ke attachment anywhere, if they ask what is the golden standard method for parasite, the answer is microscopy technique. Sebab tu hingga hari ini, most of the parasite lab, they still rely on microscopy. Tapi apakah the weakness? You really need a good microscopy. Kena duduk berjam-jam baca-baca slide. Selain tu, tapi untuk blood macam malaria ni sekarang dah ada mesin dah. Mesin akan read. Tapi untuk cacing dalam usus, parasite di usus, still we use manual method lah, microscopy, PCR and so on. Overall, golden standard is still microscope lah untuk parasite ya. Untuk virus, fungal, bakteria tu lain lah. That's not my field. But usually bakteria kita guna gram stain atau certain stain, lepas tu kita buat parasite. Kalau virus, you have to culture in cell. Fungal, you culture dengan agar. Lepas tu you can do PCR and so on. Tapi kalau parasite, you still go microscopy. Why do you think so? Mengapa anda rasa untuk parasite microscopy is still the golden standard? Any opinion? Pelajar-pelajar Yogyakarta dah tengok ke parasite di slide di masa makmal? Ada kan slide-slide yang cikgu ajar sana? FYC slide. So if you see, kalau parasite, kalau saya bagi the students any specimen, dengan mata sendiri you boleh tengok, oh ini parasite ke tak ada. Then you say yes or no. Sebab kalau you dah ada atlas yang diberi pada kamu. At the same time, kalau saya bagi bacteria culture pada you tengok dalam parasite, semua nampak macam kuning. Semua nampak rod shape. If rod shape, then you katalah, oh ini bacteria, tapi nama you cannot bagi. The name, same thing goes with fungal. Tapi, parasite is different. Di microscopy, you can actually see, oh yang ini Giardia, yang ni Trichodis, yang ni Ascaris, hookworm. Ini macam, it's very clear to your eyes. So that's why we still say, untuk parasite, the golden standard technique is microscopy examination. Okay. However, if an unequal identification of parasite cannot be made, the specimen should be analyzed using polymerase chain reaction, PCR. Like maksudnya you bacalah, you nak pergi rehat, lunchtime, you're hungry, dah terima 2 specimen, you still your heart, ini Entamoeba histolytica ke atau Entamoeba dispar. Nampak cacing macam telur macam Trichoris trichuria, tapi saiznya lebih panjang. Maybe ini Trichoris canis ke? Canis you know is dari anjing, Trichoris canis. So if you cannot make a decision, then the best thing, extract DNA and do a PCR. solve all your doubts in this. Lepas tu kita ada PCR amplified fragments can be analysed. Selepas dah buat PCR, kita boleh analyze amplicon atau aplikasi kita guna RFLP, which is restriction fragment atau sequence. DNA sequence sekarang banyak kita guna, especially zaman COVID ni, they buat DNA sequencing. Di setiap negara lah mereka ada pusat-pusat nak buat sequencing. And mereka tengoklah, oh sequence ni Delta, Corona is Delta strain, Omni strain, Alpha strain, Kappa strain and so on. Yang ni information hanya boleh diterima dari DNA sequencing. Tapi untuk every pesakit kita tak buat lah because DNA sequencing memang mahal. So we just take macam certain bandar ke, they take a few lah and they do the random sequencing. Okay, so this is something. Okay, yeah, so far. Okay. Benefit of using molecular-based assay in parasite diagnostic. Apakah keperluannya nak guna molecular-based ni? Kenapa tak guna saja serologi? Kan serologi lebih senang. You just ambil darah, spin, you put 1 drop, you already dapat results. Isn't that easy? Molecular ni cerita panjang because you kena extract DNA, reagent dia mahal, you need a machine and so on. Okay. The reason is highly sensitive, higher in sensitivity. Okay. Pelajar-pelajar, adakah kamu mengenal perkataan sensitive? Higher in sensitivity. Emotion sensitivity, I think you understand lah. Lecturer marah sikit, menangis, atau anda marah like that. So kalau di makmal, kalau kita teknik ni sensitif, bermaksud dia boleh detect. Even kalau kamu ada sikit parasite pun dia boleh detect dia. Sebab tu kita kata ni higher sensitivity. Specificity, perkataan spesifik. Spesifik ini bermaksud they can tell you, oh ini Trichuris species A, ini Trichuris species B. Oh ini Ascaris untuk dijumpai di manusia, Ascaris yang dijumpai di lembu, di anjing and so on. So specific to that kind of species. Enable to quantity. Okay. So let's say sekarang you ada pesakit. Okay, dan kita nak tahu pesakit ni dia ada berapa quantity of parasite dalam tubuh badan dia. You cannot do that with microscope. Sebab microscope you cakap okay yes or no. Tapi maybe malaria you can count lah. Kalau GIP you tak boleh count. Usually you jumpa you record yes or no. Atau egg you can quantify it lah. You guna McMaster chamber. But still if you want to know the infection real time tu, you guna PCR assay. And dia lah rapid. Even though this they are quite fast. Sekarang kita ada kit and so on. Okay, pelajar-pelajar, so far are you all okay? Any doubts? Ada apa-apa persoalan? Sorry, is there any question? Okay, you can actually type in also lah. If your mic is not very clear, you can type in also. I can answer the question, okay? Then later 3 o'clock we take a 10 minutes break and we continue. Don't worry. I know kalau molecular sini a bit dry lah, technical, so it's okay. Okay, so this is example of molecular assay. Okay, so we will see overall, then kita akan pergi ke different molecular assay yang kita tahulah, yang commonly used. Okay, molecular ini PCR, the name is polymerase chain reaction, was developed by Carl Mullis, the scientist who developed this in 1980. I know you all belum lahir lagi kan? Okay, dia merupakan seorang ahli biokimia, ya, and dia juga telah memenangi Nobel Prize, one of the Nobel Prize, but we have to be thankful to Carl Mullis. Without his invention, until now we do know what is PCR. His detection, his idea helped us a lot. Okay, just to tell, PCR ni selain daripada field of parasite, kita juga guna PCR di untuk virus, bakteria, fungal, case cancer, yes, and also importantly forensic science, case science forensic dalam case jenayah. So you know, masa case jenayah usually the pembunuh tak tinggalkan belakang banyak dia punya DNA. Jadi kita just ambil dia punya rambut, bendalir, and we do a PCR. Aside from that, dalam bidang pertanian, banyak PCR digunakan untuk GMO, nak tengok, okay, ini adakah bakar yang baik untuk padi and so on. Okay, so that is importance of PCR. So everyone actually, if you're coming from the lab base, you must have the idea of PCR. Kalau tak dapat hands-on pun, you must ada satu a bit of idea lah. So PCR is using based on ability of DNA. Deoxynucleoside. So polymerase to synthesize new strand of complementary. It needs a primer to bind. It requires possibility to delineate a specific region, and at the end of the reaction, a specific sequence will be amplified, which we call amplicon. So it's like this, student. It's not very difficult. I'm sure semua dah tahu apakah dia DNA kan? Have you all learned genetic? Dah ambil genetic ke? Ada subjek genetic? DNA, you know, is important structure, the molecular structure in our body. So kita punya DNA, dia ada double strand. DNA macam ni double strand. So apa kita nak buat? Kita nak bagi certain region, certain bahagian ni kita nak buat untuk macam beberapa kopi. Kita nak tambahkan kopi dia untuk melihat kadar infection. Okay, sekarang imagine this is ascaris lah, ascaris punya DNA. Jadi saya nak I just dapat, let's say, satu cacing saja dari sebuah kampung, tapi saya nak tambahkan cacing dia untuk simpan reference saya. So what I will do, I akan design primer untuk satu region. So first stage DNA itu PCR, kita kena bagi suhu yang sangat tinggi, 94 to 96 degrees, untuk pecahkan DNA dia. Suhu yang tinggi. Selepas dia pecah, macam ni lah, pecah bagi dia dua bahagian ya. Kita ada sequence ATCG ya, yang dia akan bind, call it forward primer, revert primer, untuk mengamplikasikan region yang kita nak di sini. Sequence pertama, you pecah dia. Sebab DNA bond memang kuat. How you break it? You put a high heat. Kepanaskan dia. Region yang ni, you kena synthesize sendiri dari company. Kita order dari industri, forward, reverse. So this primer, apa yang dia buat? Dia akan mulakan pekerjaan dia lah. So usually dia akan pergi A bind dengan apa? ATCG. Kalau A, T akan bind. Kalau sini C, G akan bind. Lepas tu kita ada ATCG nucleotide, dia akan tambah. Lepas tu kita ada satu enzim, we call it DNA polymerase. Dia akan bekerja lah. So dia akan ikatkan amplicon yang baru. Dia bubuh enzim. Then you get a lot of copies. 2 cycle. Kita ada 16 kopi, beratus-ratus kopi. Jadi from one ascaris sequence ini kita ada macam berlambak kopi, berjuta-juta kopi reaction. So this is very specific. You tengok kita adalah like say genome dia banyak. Tapi kita akan target, kalau let's say sekarang saya tak pastilah ini ascaris lumbricoid ke atau ini ascaris sum. Saya tak pasti. So what I will do, I akan ambil sequence, primer sequence, yang ni kita boleh dapat dari gene bank lah. Atau bagi suruh industri untuk synthesize pada kita. Then kita target pada satu region ni yang spesifik. That means ascaris sum ada, ascaris trichoris tak ada. And so on. Okay, so and it will be very specific. Okay, so far any question on the basic principle? Adakah anda faham? Or am I going too fast?
Faham.
Faham. So the primer usually ATCG ni kita beli dari industri lah. Kalau kita ada molecular company sini, kita just call them, kita order. Sebab yang ni usually I'm not sure about Yogyakarta, tapi di Mestek pun kita tak ada sequencer ni. Sequencer ni mission yang besar, and ini kena kepakaran untuk orang untuk buat ATCG semualah. Jadi kita just order, tapi mission PCR kita ada. So like that. Okay, what we need. Okay, ini merupakan ingredients, bahan-bahan yang kita perlukan untuk buat PCR. First and foremost, DNA template anda, primer, ATCG. ATCG ni apa dia? Cytosine, guanine, adenine, thymine. DNA. Tapi kalau di RNA, T tak ada. Diganti dengan U, uracil di RNA. Tapi DNA, we have this. Kalau G and C, dia ada 3 bond. Kalau A and T, dia ada 2 bond saja, hydrogen bond. Sebab tu kalau kita tengok bond di, what is bond Nadia in bahasa? DNA bond, rangkaian is it?
Rangkaian.
Okay, rangkaian di antara cytosine and guanine, dia ada 3 bond, you know. Kalau nak pecah ni susah sikit. AT lebih cepat nak pecah. Sebab tu kita selalu bagi suhu yang tinggi lah. Like macam 96 semua. Which we call it denaturation step. So kita bagi mereka pecah. Okay. Then what we will do, all this. What is the function of DNA polymerase? This is the enzyme nak bagi mereka semua lekat. Macam gum lah, dia initiate the polymerization technique. Okay, kita mix, we put in a tube, then put dalam machine. PCR machine ni, we call it, the correct name is thermocycler. We have a few steps in PCR. First, kita panggil step denaturation atau step untuk pecahkan DNA bond tu. Pecah. Okay. Selepas dia bond dia dah separate macam ni. Sekarang 2 kan macam rapat, dia pecah dah. Now we have annealing. Annealing bermaksud suhu yang the primers will come and bind. Primers ialah macam first yang initiate DNA line nak dekat sini. So kita ada forward primer, kita ada forward primer, kita ada reverse primer. 5' to 3', 3' to 5'. Dia akan bacalah primer. So primer ni selalu kita ada just around 20 base pair saja. Around 20, 16. Pendek, dia tahu mana nak lekat. Dia akan dapat lekat. How dia lekat? Sebab dia tengoklah kalau primer ni ada A, mesti dia lekat dekat T. Kalau primer ni, kalau ada C, dia lekat kepada
G.
G. Good. The same thing. Dia akan go. Lepas tu, yang ni kan kita ada dalam buffer. Mereka akan lekat di sini. Dengan pembantuan tag polymerase. Then the extension started. Extension ini perlukan satu suhu, we call it 72 degree extension cycle. So apakah dia enzim tag polymerase? Okay. Ini merupakan satu enzim. If I'm not mistaken, dia is actually from a marine life, you know. Dia macam dari sea creatures, they take. Ini merupakan satu enzim yang tahan dalam suhu yang panas. Enzim-enzim di badan manusia usually dia tahan sampai 37 our body temperature. Selepas 37 degree Celsius dia memang denature, dia tak boleh tahan. Tapi Carl Lewis dah cari satu enzim, we call it tag polymerase. This is something like a miracle lah, miracle for us. Miracle enzyme yang boleh tahan suhu yang panas. Thermophilic temperature, dia tahan tag polymerase. Lepas tu sekarang dalam bidang molekular adalah beberapa enzim lain lah. Dia panggil PFU and so on. Tapi semua idea start from tag polymerase. Okay, so this is how it goes. Ini baru satu cycle. Pecah, anneal, extend, elongation. Ini akan repeat untuk 30 cycle. 30 atau 40. Actually 30 should be okay. 30, then you akan dapat produk yang berlambak. So you can see, we put here all. Benda semua ni kita bubur dalam one tube, bubur dalam mesin, run gel. Okay. Tapi nowadays, this is the asas lah, bahan asas idea you must know. But nowadays kita sudah ada cara yang mudah di mana nucleotide, buffer, enzyme 1, 2, 3 didapati di company molecular biology, semua jual di satu tube saja. So kita panggil dia PCR enzyme. So I think nowadays you all tak payah belilah nucleotide, mixed buffer, tag polymerase and so on. Semua dah 3 in 1. Your younger generation is 3 in 1 generation. Masa I learn PCR, masa tu kita kena eat satu-satu kita add. Tapi sekarang mostly the companies, and I know I read some journals in Indonesia pun dia dah guna yang pre-mix. So apa yang you kena add is just primer, DNA sample dan air. Then just bubur di mesin. So that is the advanced of all the biotech in the whole world. Tapi save your muscle. So lah kalau tak, berapa you nak pipette kan? Okay. So this is basic. Kita akan tengok various type of PCR yang boleh diguna di bidang parasite. Kita ada single PCR, This is what I explained tadi. Nested PCR. Nested PCR ni you nak terperinci saja, nak amplify certain thing. Multiplex. Multiplex ni you nak target beberapa gene dalam satu cycle. Reverse transcriptase. Quantitative. LAM assay. LAM assay ni we don't use in the lab, but this is one of the technique that I will explain lah. Okay. So before I take a 5 minutes break, let me share with you all apakah kepentingan atau how do I say? What are the important step? Step yang banyak crucial untuk PCR. PCR actually is not difficult. Tapi kalau you tak dapat results, you pening. Sebab you don't know from where you want to start. Adakah DNA saya ada contamination? Adakah enzim saya tak bekerja? Atau mesin rosak? So, you kena start dari the stepping stone should be a very good stone. That means the initial technique must be very good. Start DNA extraction kamu mesti bagus. A good quality of DNA. No contamination. Bila nak buat PCR, mesti buat di bilik PCR. Tak boleh buat dekat sebelahan dengan animal house. Atau berdekatan dengan tempat kamu extract stool. No. No inhibitors. Check patient yang kamu terima itu, adakah dia makan apa-apa ubat antibiotik ataupun ubat sembelit macam ada contain barium sulfate. Kalau yang tu semua ada, bila you extract DNA, dia akan inhibit untuk PCR. So just why MLTs yang kerja di hospital, bila dapat slip nak buat PCR tu, dia kena baca umur pesakit, clinical history, adakah dia ambil ubat and so on. Okay, so this is single PCR. Okay, shall we take 5? Shall we take 5? I mean, shall we take a break at 5 minit? Rehat 5 minit?
Boleh.
Boleh. Okay, so we call it take 5. Okay, just take 5 minutes, then you'll come back.
Baik, thank you so much for the material, Dr. Mehru. Sebelum kita menunggu moderator, mungkin kita akan langsung saja masuk ke sesi tanya jawab ya.
Actually, I have not finished. I just nak ambil bagi rehat 5 minit.
Oh, ya.
Okay, okay, just a few minutes. Okay.
Sorry.
No worries, no worries. We always stop after usually 1 hour. I'll give 5-minute break, nap and washroom break and so on. Okay, we'll continue now, students. Okay, so we shall continue. Okay, so as I said, kita ada beberapa jenis PCR, single, nested, multiplex, reverse transcriptive, quantitative, and LAM assay. Jadi kita akan tengok one by one lah, what is the different session of this technique and kegunaan dia di bidang parasite. Okay, this one I explain. So single PCR, as I mentioned just now, this was the first technique designed by Carl Lewis. Bila dia dapat, oh ya, kita boleh buat PCR. So this is actually the simple, we call it a single PCR technique. Di mana kita choose target DNA. Sekarang saya ada PCR, let's say sekarang trichuris hookworm pula. Ada, okay, tak mau worm pula, kita choose yang lain. Okay, Giardia. Kita ada Giardia, tapi I nak tengok dia Giardia lambia ke, duodenalis ke. What I do, I extract DNA dia dari stool sample. I cari primer yang spesifik untuk Giardia duodenalis di gene bank. Kita boleh synthesize. Secondly, kalau tak mau buat sendiri, kita boleh cari ambil primer di mana researcher lain yang sudah guna di kajian mereka. Yang ni pelajar-pelajar boleh cari dari jurnal lah, baca jurnal, primer interest yang orang lain dah guna untuk diagnostik, kamu boleh guna. It's no problem sebab primer je. Tapi untuk diagnostik lah, just for diagnostik, you design primer, buat PCR. Bila kita dah buat PCR di tube, ini machine, okay, dah habis, you kena prepare the gel. Okay, ini we call it agarose gel lah. Di gel ini kita akan load the DNA. DNA kita adalah charge negatif. Jadi it will move to the positive charge. Kalau size DNA amplicon kita just beberapa ratus base pair, dia akan cepat lari. Dia kan smallest. Tapi kalau dia besar, genomic dia besar, dia tak boleh gerak gel ni. Jadi dia akan slow, dia akan berhenti di sini. Usually this is a large base pair lah. Macam 1 KB, 1000 KB ke 2000 KB and so on. Okay. This is the one which we know. A DNA template, how the primer bind. As I explained before, di slide di dulu. And tag polymerase. Tag polymerase lah yang enzim yang mujahid can actually help untuk PCR ni bertambah. Okay, so this is single PCR. Kedua, we have another one called nested PCR. Nested datang dari perkataan sarang kan, nest. Tapi nested di sini bermaksud is a modification on single PCR. Yang ni kita guna untuk meningkatkan sensitivity atau specificity of our PCR product. Apa beza dia? Yang ni kita akan guna 2 primer. Kalau students tengok sini just 1 primer, forward reverse. Yang ni kita akan guna 2. This has been used dalam bidang malaria. Pernah guna, they have also used example lah. Okay, how it works? Okay, andai kata ini adalah malaria knowledge dari monkey species kan. Okay, you ada dah. So you put primer A, primer B. Bila buat PCR, you dapat yang besar ni, junk yang besar, amplicon besar. So yang ni dapat sini dah. So ini pelajar punya amplicon, results dia kan. Lepas tu, you akan guna template ni sebagai DNA. Okay, first yang ni you dapat dari kit, you extract DNA. Lepas tu you buat PCR, you dapat result ni. Result ni akan digunakan sebagai PCR yang kedua. Guna primer yang kedua. So you have 2 primers here. So kalau tuan-tuan dan puan-puan lihat di sini, you akan terperincikan. So you akan actually concentrate the amplicon. So that is why we call it nested PCR. Terperincikan dia punya results. Okay. So you all understand this? We use 2 primer. So how many primers?
2.
Congratulations. Yeah, you understand it. Okay. The next, we call it multiplex PCR. Okay. Multiplex. Dari word, this word, perkataan ni, multi ialah apa? Berlain-lain kan? Multitasking, multi-usage, more than one. So what we do is, that means multi sini kita guna beberapa primer. Kalau nested 2. Yang ni are designed to amplify more than one target simultaneously. Kita tak target satu region saja, kita target beberapa region di strain itu. Jadi kita guna beberapa one or more than one primers. And also kalau DNA itu kita ada beberapa DNA yang boleh tambah dalam tube. Okay. Sekarang student may tanya untuk apa kita guna ni doktor? Kalau tengok yang single nested, dia akan ambil masa yang lama. Sebab kalau you ada ascaris saja you boleh buat. Lepas tu you dah habis ascaris, kena extract, kena guna mesin untuk hookworm pula. Mesin tu you tak dapat share lah, sebab kalau ascaris dah buat 3 jam, lepas tu you kena run untuk hookworm. Dah buat 3 jam sampai pukul 6 malam, 7 malam you buat untuk trichuris pula. Tapi in multiplex is different. Dalam satu tube ni, you boleh bubuh strain A, stool B, stool A, nak detect beberapa spesies. Beberapa target yang interest you nak tengok. Kalau stool, 5 stool saya ada, saya bubuh dalam. Tapi ini untuk not on diagnostic lah. Atau stool yang sama I guna, strain yang sama, tapi I nak detect beberapa band. Save the time. As they have used multiplex PCR untuk intestinal parasite infection in a control trial. Okay, that means dia ada, let's say dia pergi ke perkampungan ni, dia ambil najis 5 pelajar, 5, 1, 2, 3, 4, 5, 5 lah. 5 pelajar ni I nak detect, sebab saya nak save masa kan. So I nak detect trichuris, ascaris, hookworm, giardia dalam satu botol ni. So apa I will do, I kena bubuh 5 jenis primer. 5 primer. So dalam satu kolom ni dia boleh detect, oh at one shot lah, tak payah buat separate. Multiplex is very good, very novel. Not novel lah, dah guna dah, but the challenges in multiplex is you must know how to optimize the primer. Sebab yang ni semua bekerja dengan temperature. Setiap ni ada temperature dia sendiri. Suhu dia you kena pandai optimize dia. Dia akan ambil masa yang agak lama lah nak optimize. Kalau dah optimize, then straight away you boleh cepat buat. So it is save your time. So how many target DNA and expected in multiplex? How many target? Several targets. Several. Many, not only one, several targets. Okay. The third, you call it reverse transcriptase. Reverse transcriptase PCR. Kalau pelajar tengok, PCR ini adalah DNA base. Kita ambil DNA, lepas tu kita buat PCR. Tapi ada setengah kemungkinan apa yang kita extract ialah RNA, bukan DNA. Why kita ambil RNA? Sebab berbanding dengan DNA dan RNA, RNA dia bersih sikit. Dia tak ada macam intron. DNA ni ada intron. Intron bermaksud intervening sequence. Sequence yang suka kacau lah in between the DNA. Kan DNA ni panjang, so macam adalah macam sampah di sini, dia jumping lah, Tapi RNA dia bersih. Sebab selepas RNA kita akan pergi ke protein. So dia ni bersih sikit. So what we need, setengah kajian kita kena guna RNA bukan DNA. Tapi ini kena ditukar balik ke DNA. So that's why the name is reverse transcriptase PCR. Reverse bermaksud dari RNA to cDNA. You reverse dia balik. Selalu macam ni kan, DNA, RNA, protein. Sekarang dari RNA you tukar balik ke cDNA. Ini ada beberapa bahan kimia atau kit kita akan guna. Lepas tu kita buat PCR guna cDNA. Okay. So reverse transcriptase, or we call it RT-PCR, allows the use of RNA as a DNA template. And addition steps allows detection of application of RNA. the quality and purity of the DNA template, of RNA template is very essential. Okay. So reverse transcriptase also has an RH function. That's the enzyme which degrades the portion of hybrid RNA. That means RNA kalau dia bila convert tu yang tak perlukan tu, it will just throw away lah. So the challenges in this, kalau anda tengok DNA dia stabil sebab dia double bond. You can even kalau terlupa letak di bench, tak letak di -20 pun, next day you come, it will be stable. Even after 1 week, it's still okay lah. Tapi RNA, nak kerja dengan RNA ni, it's very sensitive. RNA is single-stranded and they tend to degrade very fast. So RNA, if you want to work, you have to be very careful. You have to use RNA's cleanup kit and so on. So that is the difference. So reverse transcriptase PCR or RT-PCR uses what as template? Uses what as template, students?
mRNA.
RNA, yes. mRNA or RNA as template. Thank you very much. Okay, glad you all are understanding. So the only different, kalau single PCR guna apa? Single PCR guna DNA as template. Reverse transcriptase use mRNA as template. Template which actually you tukar balik pada cDNA. That's the difference. Okay. Last but not least, not last, okay, one more thing is quantitative PCR. Quantitative PCR atau kita panggil real-time PCR atau qPCR, which is very widely used now during the COVID time. This one is used to detect, characterize, and quantify nucleic acid for numerous applications. Dalam semua PCR yang I ajar ni, the top is still qPCR, which is yang ni lah. Okay. The bezanya, yang ni you guna machine and result dia akan keluar di screen. Akan dipaparkan di screen. Oh, berapa dah naik amplicon dia and so on. We have one unit dekat our university also. As in standard PCR, DNA is amplified. Biasa, the process same, ada denaturation, annealing, elongation. Perbezaan dia, quantity PCR ini, you akan gunakan satu fluorescent dye. Fluorescent dye, like cyber green, very safe. Fluorescent dye ni, dakwat ni akan pergi, it will go and attach to the ATCG. Amplicon dia, jadi you akan dapat macam chelating dye lah, dia akan blink. So graph dia setiap kali dia pergi attach, dan you akan dapat peak graph dia. In dye base, typically green, fluorescent label allow quantification and amplification. So based on the dye system ini, you akan dapat amplicon. During each cycle, the fluorescent is measured. So this is what the machine will measure and give you a real-time results. Sebab tu yang ni top technique ni. The only thing is this is a bit expensive. The fluorescent signal increases proportionally to the amount of DNA replicated. And this is called as real-time. That means each time, setiap kali yang dye ni pergi lekat pada your amplicon, sekarang you ada dah 10 asterisk gene lah, the ampli. Yang ni dia akan pergi lekat pada 10 gene ni dia akan bagi warna terang. Lepas tu semakin dia PCR berlari, let's say dari 10 you dapat 20 gene as keris. So lebih terang dye ini, lebih terang. So then you get a results. Okay, so this is how it work. So ada dua cara. Okay, one is Q-time ni kita boleh gunakan dalam ni ada bahagian dua lah. Yang qPCR ni ada dua teknik. Either you nak beli yang fluorescent dye yang I explain tadi. So dye ini dia datang dengan color lah, PCR mix. You just masukkan dalam PCR reagent dan lari PCR seperti biasa. Result dia akan dipaparkan di mesin. Anda tak payahlah macam load the gel, do a PCR gel, run gel, tidak perlu langsung. You just get the result from PCR machine. Sebab graf dia akan keluar di mesin. Yang ni pula more specific, lebih baik. Ini kita gunakan DNA probe. Bermaksud you have to synthesize probe yang ni spesifik, dia akan lekat pada DNA template anda. Kalau dia tak jumpa your DNA template, this probe takkan lekat. So dia akan cari until dia dapat DNA template you, baru dia lekat. So that's why dia kata yang ni lebih spesifik. Sebab bila dia lekat, dia akan result keluar lah. So kalau nak pergi more detail, we have two dye lah, quencher, fluorophore and also Taq polymerase, excited dye and so on. So this a bit more technical lah, how macam mana dia bekerja. But overview, this is the idea. Any questions? Adakah apa-apa persoalan dari tuan-tuan dan puan-puan? Adakah anda memahami PCR? Any questions, students, from your side?
Not yet, Miss.
Okay, so far are you all okay, students?
Okay, Miss.
Thank you. Okay, so we will move on. Okay, so last but not least, this is the technique which called LAM assay. LAM assay technique. Okay, LAM assay technique was developed around 10 to 15 years, if I'm not mistaken, ago. But the usage in parasite is very less. Okay, the reason mengapa ya teknik ni, I think dia start dari Japan teknik, dari from Japan. Yang ni, kalau pelajar lihat, untuk PCR ni kita memerlukan enzim yang mahal, bench yang bersih, mesin diperlukan, pipette and so on. Tapi macam mana I nak tengok kalau I nak buat essay ni di tempat kerja saya, like on the field side. Let's say I'm travelling, I travel dekat satu kampung, I nak ambil sampel, I nak buat result di sana, on spot I nak test, tu sakit ni ada so on or not. So this is what LAM assay is all about. LAM assay ni dia gunakan beberapa jenis primer, 4 ke 6, dicampur di satu tube, tapi dia amplikon dia sangat panjang, dia pergi beberapa kb lah. Tapi yang susah sikit yang di sini ialah primer design dia. Ini kena kita bekerja sama dengan company, kita tengok dia ada satu software untuk buat primer. Kita campur primer, sampel najis, enzim di dalam satu tube, and kita letak di mesin. Yang tu kita boleh bawa di kereta jugalah. We call it hot block. And kalau tube tu bertukar warna, then we say it's actually positive. So these are some of the explanation. A stain replacing DNA polymerase initiate the synthesis of 2, especially from the loop. So the primer will go here and synthesize. Lepas tu akhir sini dia akan buat macam loop, synthesize. Lepas tu akhir sini dia akan buat loop. Yang loop yang ni sebab tu dia datang, kita dapat strand yang sangat besar, size dia besar. Okay. So DNA products are very long, around 20 kb, and formed nucleic repeats. So you have a very long DNA. Okay. So example, this is the procedure. Okay, you do a DNA extraction. Okay, tissue sample, lysis buffer. We mix it on the spot. Okay, then you put the enzyme. Okay, kalau dia tukar, yang ni kalau dia tukar macam biru, it's positive. This one negative and this one control. So kit tu akan beritahulah, our stool kit atau lime estate kit, dia akan beritahu kalau tukar warna apa, ialah positif. So just why yang ni digalakkan sekarang sebab kita boleh buat at the spot and tidak perlukan mesin yang mahal. So this is actually one of the procedure, tapi frankly in bidang parasite kurang diguna. Hopefully in future we will investigate this more lah. Okay, so I think that's all the technique from me, and I'm going to share with you a video which is done by Miss Nadia, my master student here. How do we isolate DNA? We run a PCR in our lab. So you will have a better idea. Okay, before that, thank you very much. And you can see this is our work that we are doing with parasite student in Mestek. This is my student Nadia during their visit ke kampungan orang asli. Di Malaysia kita ada puak yang orang asal di Malaysia, we call it orang asli. Or Aborigines indigenous community. So I'm not sure kalau di Indonesia what do you call macam dia macam orang pribumi lah. Mereka ni they stay near the forest region because they are very close to the nature, and they have lack of sanitary facilities, kurang tandas macam tu. So they always have parasite infection. Kalau kita tengok sini, my student dia akan bagi ubat central lah. Usually bila bagi ubat ni kita kena directly go, directly we give them. This is my first master student who worked in one of the village, and me and my students, we are actually in one of the competition. Okay, so before that, I would also like to welcome the students from universities Yogyakarta, and I would like to take this opportunity to inform that our university, UNICAL Mestek, we have a conference coming in November. So the price for international student is USD 50 and the others as here. So I would actually welcome all of you to join if possible. Sebab the price I think is quite cheap compared to other conference. It's an international conference. Kita ada CPD points kalau sesiapa nak publish di journal and so on. Okay, so at the same time, let me share our website. Okay, so this is our conference website. If you all can see, it's on the way. You all can explore. The registration link is here. So please, I will send also an official information to your university to tell about our conference here. Okay, so with that, thank you student. Last but not least, let me share with you the video on how do we do a DNA extraction. Can you all see this?
Yes, we
Okay.
can.
So I will explain verbally, because the video, I think Nadia didn't talk much, but I'll share it here. Okay. So we will start from our location. Okay. Kalau student tengok, ini merupakan site yang we go to the orang asli and take. Kita ada a lot of contaminated soil. And as I said, the population there, they prefer open defecation or near the bushes because that's their culture. Okay, we have a lot of wild animals, dogs, and so on. So the soil memang contaminated. So around there. So we ambil juga soil, kita ambil sampel najis and kita bandingkan. Sebab kalau kita tengok memang parasite ni datang dari air, soil dan human sendiri. So ini merupakan bahan-bahan yang diperlukan bekas untuk ambil najis. Yang ni kita ambil dari orang asli lah. Kita akan bagi hari pertama dan hari maybe kedua atau ketiga, we'll go and collect from them. Okay. So when you already get a soil sample, the first thing you must do, you must kacau dia betul-betul. Once you dah mix, dah kacau dengan baik, then we will do a filtration. Filtration ni penting untuk mengasingkan apa bahan-bahan yang besar ada di najis. Easy for us to extract the DNA. And you can add some sodium chloride lah. Kalau dia pekat sangat. Ini gauge biasa ya. Okay. So selepas dah prepare tu, so now we have a stool sample, filtered stool sample. And we will use a DNA extraction kit to extract the DNA. Kit ini, it depends on us. You can buy any kit. Usually what we will do is we will test with a few kit. Whichever gives us the best yield, we will buy from that supplier. DNA extraction menggunakan method kit. Kita ada beberapa proses. Pre-lysis. That means kita akan lysiskan DNA and also benda-benda besar di sampel najis. Dan kita akan tambahkan proteinase K to digest the protein. As we know, sampel najis ni semua ada kan, banyak-banyak sampah sarap. So many things we have. DNA, protein, sometimes undigested food materials and so on. Then you put a binding and washing solution, dilution. Let's see. So this is also a very important step, pre-lysis and cleanup. We have to use a very clean pipette tip after autoclave. Despite it's a stool sample. So just why you kena filter dia. Kalau tak, pipette tak boleh masuk dalam lah. If a lot of things are there. So this part is a bit difficult lah dengan stool. Some stool yang kalau pesakit tu ada sembelit and so on, very difficult to pipette. So you need to put sodium chloride to dilute the sample. Okay. Okay, next we use the lysis solution. Lysis solution is mainly to lyse all the macro items, like kalau undigested, bagi dia najis tu jadi benda yang ringkas dah. Dia ada beberapa chemical inside as provided by the manufacturer to make it simpler for our extraction. Yang pentingnya bila buat spesimen persediaan ni, you kena vortex dengan bagus. Mix dia very well. Okay. So once dia dah ready, okay, kita akan transfer dia to tube macam ni, which provided by the supplier. Tube ini kita collect, bawah ni kita panggil dia collection tube. Yang atas ni, kalau student tengok, ini adalah membrane filter tube. Membrane filter tube ni very important sebab dia tak bagi yang benda-benda yang kasar to go down. They will perangkap here like a filter. Okay. So once you put, you have to centrifuge and add protein digestive K. Proteinase K ni you must keep in chiller, lalu selalu kena disejukkan. Then we do our washing and binding process. Washing and binding solution ni we use alcohol. Dia memang ramuan dia is already mixed with alcohol. A few times you kena wash. Okay, now we are putting in the collection tube yang ada membrane ni. So we estimate DNA yang stool sini sudah really bersih sikit lah. We just want to memerangkap the DNA in the membrane filter. Okay, so a centrifuge is needed. Again, a few times we wash. Okay, so proses yang DNA extraction ni you will need a few times of centrifuge. So what she's explaining is the final wash solution, you will actually get DNA at the end of the tube after using the elution buffer. Okay, so this is the elution. So elution buffer towards the end, dia akan tolak DNA kepada tube. If selain elution buffer, you can also use water lah, distilled water. Also serve the purpose. So example, the DNA we get here. Lepas tu DNA yang kita dapat ni, kita boleh quantify using a machine, we call it Tecan plate reader. Okay, so sekarang dah habis. DNA extraction dah habis. We will see untuk PCR pula. So as I said, PCR, sekarang kita ada master mix and kita akan add the DNA dan air. That's all. Okay. So usually the DNA is here. Okay, extracted DNA tadi. Kita ada water, nucleus free water. We need a very clean water. Okay, enzyme. Yang ni dah mix dengan ATCG, nucleotide and so on, premix. Primer, reverse primer. Today she's doing for ascaris, and reverse forward, ascaris forward primer. Okay, semuanya kita kena buat on ice. Very important. So bila dah bancuh, mix, we have a few formula. So masa ni it's difficult to record. You need to do it quite fast. So it looks like this. So bila buat PCR, tuan-tuan puan-puan make sure ada satu control, satu negatif, satu positif. Very important. For 3 tube, minimum 3 tube lah. Positive, negative, and control. Your sample, negative control, positive one, and sample. So this is our PCR machine dekat our university. It's an old machine, but it works very well. And we'll put at the tube like this. Okay, so then you press run. Like for me, I have my students have it under my name. So the initial temperature that we use is actually at 94 degrees. Then it will run, depends on which PCR machine you're using. We set at 30 cycles. So this PCR will take around 3 hours. Like sometime 1 hour also can. Tengok berapa size amplicon yang kita perlukan. Selepas itu kita, let's say now PCR dah habis. Sampel-sampel itu perlu di spin. You have to spin it to pull it down, untuk tarikkan semua fluid di bawah. Okay, sekarang PCR dah habis. DNA dah buat, PCR dah buat. Sekarang kita kena run gel. We want to run the gel and see PCR kita ni sukses atau tak sukses. Kalau sukses, you akan dapat band yang amplicon tu di agarose gel. So yang ni kita kena prepare agarose gel lah. So agarose gel ni ada dua, kita boleh buat either TBE, tris boris acetate, atau TAE. In our lab we use tris acetate. So how we do? We just mix it, bagi dia 1%. 10% we make it 1%. We call it one time TAE buffer. From 10 time we make it 1 time. Then you masukkan serbuk agarose. Untuk 1% gel, we kena perlukan 1 gram agarose di 100 ml of TAE buffer. Then we mix red safe. In our lab, I don't use ethylene bromide. Ethylene bromide is a gel that it's a dye that we need to use to observe the DNA. But in our lab, kita tak guna. We use red safe. Sebab ethylene bromide is carcinogenic, so we don't have a separate room. So we use cyber green and replacement is red safe, which is very safe. This is not carcinogenic. We just mix in our gel to visualize the amplicon. So just put a few drops, just 1 drop, very minute. to be red in color. And you have to mix well. You have to mix well and you pour in our gel cascade. So yang ni is comb. We call it comb. This is the gel cascade. Di comb yang ini, we will load our DNA. Our ni marker. Macam pembaris lah, ruler dia akan tengok, oh ini 100 kb, 200 kb and tengok. And we will put here is our DNA product. Kita akan run dia. Okay. So next we have to transform it into our electrophoresis gel. So for this we have to fill the tank. Isi tank ni sampai dia punya buffer melintas dia punya agar. If not the current won't flow. So you have to make sure it's sinking lah. The gel should be sinking into the buffer. Okay, so you know that I hope students by now you know our DNA is negatively charged. So when you run the gel, the amplicon will move from negative to positive. So make sure you load the DNA here. DNA will move to the positive rod. So whenever you load, you have to practice this a few times. Gently, precisely go into the well and load it. Load everything. So this is how it looks now. And we will be covering it and run the gel around for 90 volt, maybe 30 minutes, 40 minutes. Depends on your size of the product. This one you can monitor because the blue dye will be moving. So if you feel the blue dye dah kat sini, then I think you just stop it, then you view the gel. So last, there comes the final of the PCR work, which is actually to visualize your gel to see whether our PCR is a success or not. So for this, we need to use a gel analyzer, or we call it gel imager. So this is a gel imager we have in our university. You put the gel inside and you visualize it. So bila kita nak visualize, you expose to UV light, automatically it will happen inside here and you'll get the results. Okay, so good. We managed to get Ascaris amplicon here. So you see this is 100 base pair. This is macam pembaris lah, ruler, 100, 200, 300 and so on. And we got, so we can say our PCR is a success. There will be many times that you don't get anything. So it could be something went wrong. Your DNA not enough. PCR enzyme expired and so on. So but at the same time, if I see the band is quite thick, most probably I think the student has put too much of DNA inside here. So next time when I do, I will just subsequently reduce the amount of DNA. Tapi yang penting ni, the negative control, there is no amplicon, is clean. It shows that I don't have any contamination. So even the percentage of gel, you can change according to your need. So that's all from us today. I would like to again to thank Miss Nadia for doing this wonderful video. That's all, student.
Yes, thank you very much, Dr. Nisa, for the brief presentation. Before we end this meeting, let me read the resume for today's material. First, after that, we can move to the Q&A section. I think the students from Yogyakarta have a lot of questions to you, Dr. Nisa. Are you
Great.
still have a spare time with us?
Sure.
Oh yeah, okay, thank you so much. Okay, firstly, I would like to read the resume. Parasite is an organism that lives in a host organism and get the food from the expand of its host. Secondly, the common techniques involved in parasite diagnostic are microscopy technique, molecular-based assay, serology assay, and culturing method. But mostly parasite detection today is still using microscopy examination as a gold standard. But nowadays, molecular-based is used as a best assay because of their higher sensitivity and higher specificity, and are enabled to quantify the parasite count. One of the molecular technique is PCR. PCR need crucial preparation such as quality DNA, no contamination, and no inhibitor. There are various types of PCR. There are single PCR, nested PCR, multiplex PCR, RT-PCR, quantitative PCR, and each type has their own design and their own procedure. In the future, we hope that molecular diagnostic technique can be in the next generation types for diagnostic parasitology infection. I think that's the resume. Before we continue to the next session, we are deeply sorry, Dr. Nisa, we have no proper introduction in this meeting. So I think our students have a lot of questions. There are a question, you can raise your hand on the Zoom feature. And you can open your camera and your microphone. Please.
Thank you so much, Putri. Looks all are very well versed already with PCR. Glad to hear that. Okay, if you don't ask, let me ask you. Compared to all the technique of PCR that I show you, which you think you would choose if I give you a sample to identify Ascaris, perhaps? Okay, the video that we show is actually for conventional PCR, which involves DNA extraction, preparing of the gel, loading, and so on. So which technique of the PCR that I taught you, a few techniques, which you feel is the easiest?
Single PCR.
Single PCR, so which is actually showed in the video. So easiest, yes. But if I want to know the quantity, how much of DNA amplification is there and so on, which PCR would you choose? I don't want to count the parasite. What I want to do is I want to directly to know how much of amplicon I have.
Quantitative.
Yes, qPCR. This is, in summary, this is the PCR that you use. So in a summary, I also would like to tell, as how Putri said, I might have shared all these things, but now very commonly, directly, the students are trained to handle quantitative PCR and also single strand. If in single PCR, you don't get your results very well, then we have to use the, if you don't get the results very well, you have to optimize this in very well until you get in quantitative PCR. Okay, because quantitative PCR is very sensitive. You have to optimize. You should get some result in single, then only we'll move into quantitative PCR. Any other questions?
From Medical Laboratory Department, is there any question? Oh, okay, Mas Alif Rahman Depaha. Okay, please open your microphone, Mas Alif, and you can ask the question.
Okay, thank you so much. I want to ask something. As we know before,
Yes, go ahead.
viruses and bacteria can cause pandemic in the world, such as COVID-19, Ebola, and so on. So is there any chance for parasites can be the next organism become a global threat for the human being?
Okay, thank you, Rahman. So students, what he asked is more from bacteria, right? Bacteria virus has been recorded as a pandemic. It is like you even go to the MCO, which involves a lot of death, recognized by WHO and so. So his question is, can parasite go to that extent causing pandemic?
The next pandemic.
The next pandemic. I would assure you it will never happen. Okay. The reason is because if we look at parasites you can control it. Okay, if you're talking about gastrointestinal parasite, if you have a proper sanitation system, you can control it. And if you're looking about the vector-borne disease like malaria, filariasis, leishmaniasis, Chagas disease, African sleeping sickness, all these things, if you can control the vector, you can control the disease. But virus is different. Virus, it comes from human to human communications, from their droplets, saliva, and so on. So it's different. The mechanism of transfusion is different. Getting infection is different. Parasite is totally different. Because parasite, we know very well the life cycle of the parasite. If we can control and our treatment is still working, I wouldn't say it's not working. The only thing sometimes resistance has come and we can control it. It wouldn't go to that extent. Parasites in the colon, I know it won't go until that endemic level, but malaria, perhaps yes. But in certain region, like in certain African country, it's still endemic. And if you look at the nature of malaria parasites, the mosquito is what? Anopheles, kan? So that Anopheles mosquito, they only lives in region of forest region. They don't come to the urban side. So if we can do a good vector control at this rural area, especially targeting the forest region, so malaria parasite can be controlled. So because it comes, the infectious disease, it comes again to see mechanism how you are getting the infection. Kalau COVID is more to their droplets and so on. Okay, thank you.
Okay, thank you so much for the answer, Dr.
And I think you should be happy that parasites won't go to endemic and pandemic, yes.
Okay, thank you so much, Mas Ali, for the good question. Is there any other questions?
Yeah, anyone from Mestag, you can also ask. You don't have to feel shy.
Maybe I have, oh yes, there is a question in the chat
box. Oh,
yes.
Okay, wait.
Let me read it. Okay, in the presentation of the material, it was stated that the LAM assay method was being developed. The question is from the way of reading the result based on the color change of the homogenization result of the primers and the sample being compared with the reference, whether this method has a higher specificity, sensitivity, and accuracy values than some previous PCR methods, so that this method was developed regardless how it works in.
Okay, thank you very much, Roshida. So as I said, LAM assay is still under investigation. Okay, this was the latest technique developed, not so much, but there are still research ongoing on this technique, whether it's sensitive or not. As you said, yes, sometimes people think, oh, why is just a color changes? Am I confident it's really positive or not? But it goes back to the primers that you use. The primers that we use in LAMP assay is very sensitive. We put around 6 to 8 primers there, and they keep on forming a loop. If you don't have the right DNA, they won't form. So we call it as a failure. Maybe the end product, you just say whether the tube change color, yes or no, but specificity and sensitivity is high. But how you measure this? The primer design, if it couldn't bind and develop, you cannot get your LAMP assay developed. You won't get a positive result. So we depends the sensitivity on that part. So I understand she asking is just by color change, but it all goes back on the sensitivity of the primer developed. It looks easy, but if your primer don't bind and they don't follow the proper loop, when you put in the heating block, you won't get any results. So this is something about the LAMP assay. But I wish to explore further. I was telling Nadia, maybe one day we should try to explore this and see. I wanted to do once a long time, we want to develop in our lab, but that time it's very new and we opt for the PCR method, which is very well known. Okay, thank you, Roshada, for your question. It was very mind-blowing and it benefited all the other students.
Okay, maybe the last question. There any question?
Do you all have classes on, do you have classes on Saturdays?
Actually,
Yeah, yes, I know, right? So it's an additional classes. We do have only for flexi students. Okay. Any other question?
me. Okay, Mbak Latifah Sukma. Okay, please open your mic and you can read your question. I wanna about. I'm sorry, Mbak. I'm sorry, Latifah. Can you speak louder and clear?
My question. Mengapa ilmu laboratorium masih berpegang pada uji mikroskopis untuk mendiagnosis infeksi parasit, sedangkan ada banyak perkembangan dalam diagnosis molekuler? Thanks.
Thank you, but I think I need a translation help.
Okay, okay.
Putri, you can help me to translate.
Okay, okay. If I can translate the question. In the meantime, while laboratory science technology is still stick up with microscopy diagnosis or microscopy technique, unless there are many development in the technique such as PCR and so on. So why the laboratory technology is still stick up with the microscopy diagnosis? I think that's the question.
Is that Latifah your question? Okay, thank you, Latifah, and thank you, Putri, for interpreting. Okay, so as I mentioned, golden standard technique in the whole world for parasite is still microscope. Sebabnya it is cost effective, it is cheap. The moment you receive a sample, you take a smear, you put in the slide, it's very easy. Okay, and macam ni, okay, I give you a sample parasite, and I give you a sample bacteria yang district, let's say E. coli. Sample parasite ni, bila you lihat di microscope, you can see the shape, right? Ascaris telur, ascaris ke, hookworm ke. Directly you boleh tulis the result. That means you have an idea, okay, sample ni ada positif ke tak ada. Tapi, untuk bakteria, kita kena buat gram stain. If you do gram stain also, you tak boleh beritahu whether it's E. coli ke, Shigella ke, you kena buat further testing. Either you streak on the Salmonella Shigella agar, McConkey, and you do further testing. Then only kita ada idea yang ni. So sebab bacteria, virus, fungal, they still use other diagnostic technique. Kalau kita di bidang parasite, we have the benefit because bentuk parasite tu dapat dilihat di microscope. Kita you bubur trichome stain ke iodine, we can easily diagnose. Tapi the challenges is kalau cyst, kalau protozoa susah sikit. Then that's why we go second option is PCR. That is one reason lah, sebab is lebih mudah. Yang reason kedua, as we know, you saw kan in the video, sampel najis ini ada banyak benda contaminated. For extracting DNA, I've worked with stool, urine, blood. It's very difficult to get a good quality of DNA from stool sample. The challenges is very high because you have a lot of contamination. So that's why we still fix the gold standard method is microscope. The subsequent diagnostic method to be more specific is PCR. Even though you see Nadia doing very simply, but the maybe Nadia you can share the chances of getting a very good clean DNA is very challenging. Okay, I hope I answered your question, Latifa. Sorry, I needed a translator.
Oke, sudah jelas Mbak Latifah?
Yeah, thank you.
Oke, thank you Dr. Nisa.
Okay, any other questions, students?
Okay,
Yes, is that?
maybe question from Putri Dwi Ivanka. Okay.
My question, what is the current trend in parasitology diagnosis?
Current trend. Okay,
Yes.
so as we know, current trend depends on which type of parasite we are looking. Kalau untuk helminths, helminths parasite is still microscope. Protozoas, protozoa di usus, microscope dan juga PCR. Untuk malaria, kita guna kaedah serology. Serology like test kits and so on. For entomology, entomology is still microscope. So now I would say a combination of microscope and also PCR technique lah. So that is why importantly they want us to, I think that's the reason why they call me to share with you all on PCR. So I think PCR is an important technique lah, aside from microscope and so on. Culturing is totally different. Culturing is only done if that particular lab is doing research involving postgrad students. Thank you, Putri. Iwanka, I hope I answered your question.
Yes, thank you.
Yes. Okay. Maybe last question from student from University Kuala Lumpur.
Yes, my own student, Mastech. They all should be under with stress of final exam. Any questions from your side?
Okay.
Or Nadia, any question from you? Let's pose question. Okay.
Okay, yes. Okay. Dr. Nisa, thank you very much for your brief presentation. It's a very mind-blowing presentation because it's still a rare technique in parasitology. So I think all of the students should learn a lot about my biomolecular diagnostic. After that, let us to present the certificate to Dr. Nisa as a speaker today. Okay, please, Mas Alif, you can present the certificate. Okay, thank you very much.
Thank you very much.
Yes, thank you very much, Dr. Nisa. After that, maybe we can open our camera for a second to take a picture.
Yeah, a second time photo.
Yes.
We did earlier, so we can take.
Oke. Mas Bagus mungkin sudah siap untuk screen capture? You ready? Bentar. Sudah ya? Oke, saya siapkan ya. Oke. Mungkin 1, 2, 3, silahkan di screenshot. Selanjutnya, next page. Oke. Next page, Ken. Sudah semuanya untuk panitia sudah terdokumentasi semuanya atau belum ya? Sudah, Mas Alif? Oke. Yes. Okay. Thank you very much for your attention and for the participant. Today we have a lot of participants, and also if you want to, you can click the attendance link on the chat box. Please, you can see on the text on the chat box there are attendance links. You can click it. After that, thank you very much for your attention today. Dr. Menyelenggarakan, so all the lecturer from Medical Technology Laboratory University Department, and also all the students from both of university. Okay.
Okay, thank you so much.
Thank
Okay,
you
I
so
didn't
much.
know your lecturers were there. I was speaking in a very layman term. I hope who's your lecturers? I'm not sure who are they.
Okay. Thank you so much. And let me pass the agenda to Mas Alif.
Okay.
Thank you, Putri.
Thank you so much, Dr. Nisa.
I take over now. Okay, thank you so much for the knowledge that you gave to us, Dr. Mehru, and thank you so much, Bu Widaninggar, who has guided today's general lecture. I have the knowledge that gained today can continue to imprint on you forever. Remember, we as students are long-lasting learners, right? There is no limit for us to keep learning whatever it is. I got a sentence from a book I've learned. Struggle that you do today is the single way to build a better future. Right. Finally.
Thank you. Wonderful. Excellent.
Finally.
That's very important when you do a PCR work.
Of course. Yeah.
So those who are doing PCR,
yeah,
they know how difficult is it.
Yeah. That's right. Okay. We come to
Isn't that Nadia?
We come to the end of this general lecture. To close this event, let's close our meeting by reciting Hamdalah. Alhamdulillahirabbil alamin. Well, guys, thank you very much for a great attention of our general lecture today. See you on the next event. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much to my own student from Unical Mastech. For all the students from Yogyakarta, very nice. It's mind-blowing to see so many of you are attending here. To my own master student Nadia, and to the organizers, thank you very much. And I hope I'll see a few of you in our conference. It's actually a biomedical science conference as it related to us, our own field. A lot of people will be there. So I'll keep in touch. Okay. Thank you very much again. Have a good weekend.
Thank you very much, Dr. Meru. Thank you very much.
I hope all of you will be doing PCR in your final year project. Okay, bye. Take care.
Thank you so much. Thank you so much for all students from UniKL.
Okay, bye. Yes, thank you. The students from UniKL too. Bye-bye.